For motility, I'll come around and help you guys do wet mounts and hang drop slides, and we'll talk about some of the troubles that you can run into with these that you guys should have read about for homework, right? So you should have your homework out. We refer to exercise 311, but I have it in the PowerPoint as to the different morphologies and number of flagella you'll have. We don't do that stain on your guys' organisms. I actually tried it this summer with my STEM kids. I bought the stain and everything. We couldn't get it to work. And I did this like a long time ago, and I was really excited, right? I don't know. We have to play around with it some more. So, but once you guys figure out what organism you have, you could look it up and find out like how many flagella it has, if it has any, and what the arrangement and stuff is. So maybe we'll do that as a bonus assignment or something. Motility test you've already done. It's actually one of the tubes that's in your rack that we'll look at, and I have some demo tubes that'll help you guys control tubes and different problems that you run into with that test as well. The fluorothioalkylate medium you guys have also already inoculated. Most of you guys have yours in the rack. A few of them I have up front because I had to refrigerate them to keep them from overgrowing. And I'll explain that as we go through it. So if you're missing your FTM tube, it's because I have it. And I will give it back to you when it's time to record your results. Anaerobic jar, we don't do this one anymore because we don't have enough jars that don't spring leaks for each one of the classes to be able to do it. So we don't do this experiment anymore, but we talk about it. So you're still responsible for it. So you should have read it, and then we will also talk about it in our, I bumped my cord. There we go. In our PowerPoint here. So flagellar arrangement, as I said, and those of you guys in lecture class, you already know this, right? We already did this. So mono, or sometimes you'll see polar, trichus, referring to hair-like. How many do you think they have? Just one at one end. Amphitrichus is one at both ends. So amphi means opposite, actually. Lopho, this is a clustering. Lophotrichus is a clustering, and it can be a cluster of flagella at one end or both ends. So peritrichus is completely covered in flagella. Do you guys not have these terms for homework? Did we mess that up again? Oh well. That's okay. You guys will have the PowerPoint. And peritrichus is completely covered in flagella. Like scary cousin It from the Addams Family. It's a good visual. Thank God people still know him. Alright, so let's see. So, what do we see? Peri, right? Completely covered in flagella. And quite a few of you guys have ones that would be like this if we could stain it and see it. This is probably an electron micrograph, this particular picture. So they didn't use a special stain. What shape is that organism? A bacillus. So the next one, what do you see? Mono, just one, right? Or polar, at one end. And I know one of you guys have an organism that is like that. Now, the next two, what's the shape of those organisms? Spirillum. Or spirilia, if we're talking plural, right? Do we have any organisms in this lab that look like that? No. Right. So probably not when it comes to these flagellar arrangements. And these were probably stains where they use the flagella stain and it makes the flagella appear thicker than they actually are. These are probably light microscope pictures, these bottom two. So notice that there's multiple flagella at both ends of this organism. So this one is Lopho. But if they had just multiple flagella, just one end, it would still be Lopho. Because remember, it's a clustering. This is 1 at each end. So this one is amphi. So for a wet mount, pretty self-explanatory, right? Drop of water, add the bacteria, put a cover slip on it, look at it under the microscope. Hang drop is a little bit more complicated. Hang drop is where we actually are going to put a drop of water on a cover slip and hang it into a depression well slide. And one of the ways that we do that is we put Vaseline around the edges of the depression well. This will adhere the cover slip to the depression well, and then when we flip it over, that drop hangs. Now these are really hard to focus, but they're much easier once you focus them to visualize movement than a wet mount. Okay, so like I said, at each table I'm going to set up at least one of these for you guys so that your table will get to see, and especially for some of you guys where your motility stab is going to be ambiguous and not give you good results. They're alive, right? Because we're not going to stain them. And that's what makes it hard about focusing these as well. So what are some problems that we run into with these slide preparations? So if you over-inoculate the slide, if you have a known motile organism, say it's a control, right, you're testing out, you already know it's motile, right? If you overcrowd them, are you going to be able to see them moving? No, because they're going to be bunched up into each other. So if you don't see them moving, you would say it's negative for motility, right? But is this a true result? No, this is one of the problems we don't want to have, which is having a false result. So in this case, because we're coding it negative, it would be a false negative result. So definitely don't want to pick up too much bacteria. Right? It'll make it difficult for us to see them moving. Now, will this condition make a non-motile organism appear to be moving? No, if they're all crowded together, that's not going to make them move, right? A non-motile organism is not going to move. So if you have a non-motile, this really doesn't create any complications for you. It has no effect on your results. So, you know, if it was non-motile and it didn't move, you got lucky. Okay, sometimes they can attach to the glass slide or cover slip. If they're attached, will they be able to move? No. Or their movement will definitely be limited. Okay, so again, you may not see them move, and if it was a motile organism and it's not moving, you end up with a false negative result. There's really no way to avoid this complication. Will this make ones that are non-motile move? No. So would this have any effect on non-motiles? No. No effect. Okay. The next is a receding water line, and this happens more often when you're doing wet mounts than when you're doing hang drops. And this is where the water is moving, right? So anything in the water also moves. So imagine, right, a log floating down the river. Okay, so how would we be able to tell we had a receding water line? Because you're not going to necessarily under the microscope see the water moving. But anything in the water is also going to move, right? And in this case, fast, even though bacteria are fast, but all in the same direction. And it's very rare that bacteria are all going to be moving in the exact same direction. So that's one way to spot that you may have this complication. Now, if you had motile organisms, you saw movement, you coded it as motility, no harm, no foul, right? Really no effect. The problem is what? If you've got those non-motiles, right? So if they're like a log floating down the river, you just see that, you just see them moving, you don't recognize that they're moving fast and all in one direction, you'd code it as motile, right? But this would be a false positive result. So the first time we have a complication, it'll make a non-motile look motile. Okay, what if we're using an old culture? Are most of the organisms going to be alive or dead? Some of them are probably, quite a few of them going to be dead. Do dead things normally move? No. So, if it's modal, but we're not seeing much motility, maybe because they're what? Dead. So we end up with a false negative result, right? Because we don't see them moving, so we write negative, but this is false because they're modal organisms. They should be moving. That's why it's good news for you guys that we meet twice a week in the summer. And for me, because I didn't have to make fresh cultures for today. Right? Because the fresher they are, the more living cells, the more movement we're going to see. Will this condition make organisms appear to be moving? Again, dead ones don't move, so if it's a non-modal, really has no effect. So what do we really have to worry about? False negatives for our modals, right? Conditions that stop them from moving. And then for our non-modals, what we really have to worry about is the water moving them, right, and making us think that it's positive. The good news is that's pretty easy to spot if you know what to look for. So I have some, instead of embedding in the PowerPoint because it makes it huge, I have some videos that I've taken and posted on YouTube. And I have a new awesome one that I took last week with the kids that I'm going to be posting too, but I have it open on my computer. We're going to watch the first one. This one doesn't have a lot of bacteria, so you really got to look. And of course, they're not at the pointer, right? So I just want you to look at the field of vision as I play this video and raise your hand when you think you see bacteria moving. So I'm going to flip over to the internet. I already pulled up these links. And I'll link these to Blackboard for you guys too. I want to watch them again. And so, play. I don't need the background noise in my head. Don't forget to raise your hand, because I'll keep playing it. Alright. Here. Okay, now I'm going to start pointing them out. See this guy? It's a rod, so he's kind of flipping around. You see that one over there? I'm trying to remember where the others are. I think there's a couple right here. When we zoom in. Oh, here's one dancing. You see him? They're not easy to see. There's another one. Alright. Let me switch over to the other one. This next one, you can't miss him. Because I almost have too many. No problem finding them, right? Look at them. It's hard to focus on just one. Right? But they're still moving, even though they're pretty crowded in there. Now, I thought these videos were pretty good until the latest one I just took. So this one will give you a headache after a little while, right? You kind of have to focus just on one spot, and you can see them kind of somersaulting. Now this next one is a really big, and this one I haven't posted on YouTube yet. This is a really big, oh crud, where'd it go? There we go. This is a really big rod, and one of you guys have this one, and it forms chains. So this is, you'll see in this one, I think there's like 2 or 3 hooked together. Almost looks like a worm. It's awesome. I'm so excited. Come on, play for me. Make it full screen. How do I do that? For this one. I had trouble focusing, but it's right there. There's one all by itself, flipping around. See that? But look at this. There's one flipping. But see this one? This is, I think, 2 or 3 organisms. And this guy's a spore former, so I'm wondering if, like, the glowing inside might be the spores. See how it kind of looks like a worm? But that's a modal bacillus and more than one hooked together. It moves kind of slow. I'm trying to get him. So hopefully we might see something like that today too. Yeah, that was it. It was short, right? But you can really see it kind of inching along. I was so excited when we caught that. Okay, so who knows? Maybe we'll get an even better video today. So we do it under low light conditions, right? Because that's going to help us in the visualization. Now, one other complication you can kind of run into is Brownian motion. I know you guys had to define that for homework, right? That's the random collision of molecules, right, due to thermal energy, normal heat energy. And so this is sometimes like the vibration you'll sometimes see, okay? That's not actual movement. You definitely saw, you know, with that other video with them tumbling around, right? There's gonna be significant movement, right? If they're just like vibrating, that is just Brownian motion, okay? And I have to say I've never really seen that. And a different technique I used to use someplace else for capsule staining was using India ink. And so you get these little tiny droplets of dye. I could not look at those too long because, again, those droplets would, under heat, they would shake, right? So if you looked at it too long and the shaking, it would really wig me out. So why don't we stain them? We kill them, right? Staining kills them, okay? So if we kill them, are they going to move? No, right? So we can't stain them if we want to see them move, okay? Although it's going to help us with contrast, right? It would kill them and defeat the whole purpose of why we're looking at them, to be able to observe motility. So why are we doing this under low visibility of light? This is how we create contrast, right? So probably the best example, and this goes into the whole scientific reasoning behind, but probably the best way to explain this to you guys, and you probably even experience this with our microscopes when the light is too high and you've got too much light coming through. Also, the first time when you give a kid a flashlight, what do? They shine it right in your eyes, or their eyes, right? And it blinds you because it's just too much light at that one time. Where, like, if you're in your room and you have a night light, right? I hate night lights. It creates more shadows than if it's pitch dark, right? You have all these weird shadows, freaks me out, right? And so again, we're going to kind of be creating that kind of shadow effect by decreasing the light, right? Under those low light conditions, like a night lighted room, right, with a little light, just a little bit of light, it's going to create those varying degrees of contrast that we need. And as you saw, the cells themselves almost glow, right? Because they're going to refract more light than the background. Make sense? Okay, so hopefully we see some cool ones today. Why would you expect Brownian motion to increase? What would we say Brownian motion is caused by? Heat, thermal energy. What do we use on our microscope that generates heat? The light bulb, right? Especially these older microscopes that I prefer. They have halogen lights. Those really heat up. LEDs, not so much. But, you know, and the longer you let it sit in a room, too, they'll dry out on you, too, because of heat. And then you definitely won't see. So the next thing that we're going to look at is the motility test. So the media that we use for this, is it solid? No, it's not. And I'll send it around and I'll let you guys poke at it in a minute. And you can see that even though it stays in that tube, it isn't as solid as our agar that we have in our plates. And the reason for that is that it allows the organisms to be able to swim through. Okay? So it's what we call semi-solid, and it's going to allow them to move through slowly. So the media we use also has a special reagent added to it. And I'm not even going to bother to try and say the chemical name, but its abbreviation is TTC. So these tubes here do not contain that reagent. This is a -1. Remember, we stabbed these tubes? So if it's negative, it will stay exactly where we put the bacteria. And you can pretty much see the negative result in this tube, right? This tube is positive. The bacteria is spread out from that inoculation. So you see how it's kind of cloudy? That'd be really hard to detect though, right? In your tube, this cloudiness. So to make it easier, we use TTC, and it produces a red color when the organisms grow, right? So you can see that nice red line for a negative where it stayed just where we put it. And for the positive, you'll see it spreading out, right? You'll see that diffuse red color. And for some of you guys, you may see a little tornado, kind of like you see here, or the whole tube might be red when you look at it. The reason for that is the oxidized, soluble form of TTC is mostly colorless, although we're having some trouble with it. Notice that that tube was a little pinkish when you guys first got it. We're having trouble with that reagent. But what color does it turn when it's reduced? It's red. So you can definitely see it. And the important thing is it's insoluble. What does soluble or insoluble mean? If something's soluble and you put it in a liquid, right, it dissolves, right? If it's insoluble, it won't dissolve, right? You'll still see it in the solution. You can't get it to go away. That's the case here. It's insoluble, so it stays right where the organisms are. It can't diffuse through the media on its own, right? So it helps you see right where they are. So why is it important to carefully insert the needle, right, and create one stab line? If you kind of moved it around, you'd put the organisms everywhere, right? And then how are you going to tell if you put them there or if they moved there? You're not, right? So this would give us a false positive, right? So notice that in your guys' racks I have some bad stabs too that aren't labeled, or they might have a question mark. So this is why when you guys look at your tubes, you're going to look at it 360 degrees. You're going to spin it all the way around. Okay? Because you see how this tube, see how it's kind of fanning out right here? Look at even here. This is 180 degrees. What happened is they stabbed in here and came out here. So they spread the organism across this surface. But when you look at it 180 degrees, you can see how it's pretty flat. Right? The organisms will spread out 360 degrees. So all the way around. You won't just have one edge like this. Right? If you've got an edge like this, you got a bad stab. Or a not so good stab. Right? But do you have a motile organism? Probably not. So here's an example of one where they actually stabbed it twice. Do you see that? Needle came down once, needle came down twice. Again, see it, the flatness of it? Probably a non-motile. But again, unless you know what you're looking for, you might code this as positive. So growth will occur anywhere that we inoculate the needle into the media. So it's we spread it as we put it in, you may get the impression of motility when in fact you're not. So a single stab is much easier to interpret. How could you get false positive and false negative? Well, we already talked about false positive, right? That's if you stab it poorly. What about false negative results? Think about how what are the aspects of the test. So this is another one where it's not motile. Do you see how it's uneven? Right? This is gaps in the media where it's spread out. So again, not necessarily a bad stab, but you know, it just went in some cracks. Right? This is not uniform. So, what did we say about this media? What kind of media is it? It's semi-solid. Why? So they can move. But what if our lab technician screwed up and made it solid? Will we see him move? No. I got news for you. She doesn't mess up like that. So that's not one of the things that we'll deal with, right? So, poor technique, poor interpretation. We already talked about that. Okay. So look at this one. It kind of looks negative, doesn't it? But this really is a non-modal. What do you think is wrong with this one? Not enough organism. Not enough organism. Excellent eye. Not enough organism. If there was more organism there, we'd probably see enough that they would have spread out. So, not enough organism, or what another problem could be. Most of the organisms you picked up are dead. Right? Those are things that would stop it from moving. So, not inoculate with enough live, viable cells. Which is why, again, I made sure you guys had fresh cultures last week. Especially for inoculations. Media is too solid. Not going to be the case for us. Although as the media ages, it does dry out. Right? So that can be a problem that we run into. But mostly you got to start with live organisms and enough of them. So why is it important that TTC, when it's reduced, when it's red, it be insoluble? What did we say about insoluble? It's going to stay right next to the organisms. It itself isn't going to move, right? But when it's oxidized form, it's soluble, but it's also what? Colorless, right? It doesn't change color until the organisms change it, until they reduce it. So it's not a problem because, right, there's not going to be any color associated with it. It's only when they grow, they change it, and it's going to turn red. And then it's insoluble, it stays put. We also want it soluble when it's oxidized, so it's all throughout the media. So no matter where they grow, they'll come in contact with it, right? And then they're going to cause it to become insoluble. It'll stay right where they were growing. So remember, each time, think about the scientific method. Why are we doing these things? How do we perform it, right? What are the accurate steps for performing? What are type of results that we're going to see? And how do we interpret these? What does this data mean? So, when we get done, we'll look at those tubes and compare them to yours and determine your results. You'll also note on your data sheet for your unknown, I ask you to draw it in 2 dimensions, right? Turn it 180 degrees to make sure that it is a flat line, or if you had some V-ing that you indicate that, right? The next test that we'll look at, and as I said, for some of these I had to refrigerate them, and so I'll have to give it to you if it's not in your rack, is the fluid thioglycolate media. So what does the sodium thioglycolate and L-leucine in this media do? It creates a particular type of environment. It removes oxygen, right? So this removes free oxygen from the media. So what type of environment are we trying to make? An oxygen-free environment. What's the name for that? Anaerobic. Also, this media has a little bit of agar in it. So again, this would be classified as a semi-solid, not quite as solid as our motility media. And the reasoning for that is to slow the diffusion of the oxygen into these tubes. Because these are not sealed up tubes, over time the oxygen is going to diffuse in. Now, can you see oxygen with your eyes? Can you see oxygen gas? I'd be impressed if you could. So you'll notice in these tubes, when I first gave them to you, I had a nice thin pink line. Right? That's where the greatest concentration of oxygen is. And then it decreases as you go down the tube to just about nothing. There is an indicator called resazurin in these tubes that reacts with the oxygen, and it's going to turn that pink color you see. So if you guys look in the demo recs that I have for you, I actually have double FPM tubes, so it takes up the whole tube. And you'll see one of them is labeled uninoculated control. And you'll notice the top of the tube is what? And what's the bottom of the tube? It's kind of a yellow color, right? And the reasoning for that is that oxygen has diffused about halfway down that tube. Those are the tubes from last week. I just let them sitting out. Oh, actually, those I put in the incubator as my negative control, as it says, uninoculated control. No bacteria in there over the weekend. Did you guys see that one that she was holding up? So is it oxidized or reduced in the presence of oxygen? It becomes oxidized. Remember, oxidation is the loss of electrons. Reduction is the gain. Remember oil rig, you won't ever screw them up. So it gains electrons from oxygen, becomes oxidized, and it turns pink. And as these tubes sit, more and more oxygen is going to diffuse into them, to the point where by next week, probably that uninoculated tube, the whole thing will be pink. So one of the things I did last week to prepare these tubes for you guys is I boiled the oxygen out and then let it slowly diffuse back in, so you guys had just a thin layer. We can only do this once to these tubes, and then we start producing toxic stuff that we don't want to mess with. Right? So I had to set aside the ones that I've already boiled once so I don't boil them again. So, why is there a colored band? So we can see where the oxygen is. But the main reason why we are using this media is to create an anaerobic environment. We want to know if your organism can grow anaerobically. So basically at the bottom of your tube. So, do we want a thin layer or a thick layer of pink? We want a very thin layer, right? We want a large anaerobic environment for your anaerobes. But we want some pink so that you can see if the organism grows anaerobically. And in some of your tubes, you might even still see that pink layer, and you'll see that it's gotten thicker because, as I said, over time, the oxygen's going to diffuse in, depending on how much growth you have in your tube. So remember, right, we want anaerobes to grow. Because of that, we want to create mostly an anaerobic environment. So here's some examples of the different growth patterns you will see based on the different oxygen tolerances and requirements of the organism. So the 5 different classifications that we have. So a strict aerobe requires oxygen, so it only grows at the top of the tube. So you see tube D, you see there's only growth at the very top. If you look in your rack, which tube is the one that only has growth at the top? A, B, C, or I think it's just A, B, or C. Which one? B. B? B? Please say yes. You guys looking? Did you guys get a rack? No. Who doesn't have a rack? Because I see one up front. You guys. Yeah, go grab your rack. I missed you guys. Yeah, different ones. Okay, a strict anaerobe means it grows only anaerobically, only under anaerobic conditions. So where do we expect to see the growth in that tube? Only at the bottom, or only in the anaerobic zone. So which tube in this picture would that be? C. Right? You see how growth is in the anaerobic zone? We know where the aerobic zone is. It's the pink zone. You don't see any fuzziness there, do you? So this one's only growing anaerobically. We don't have any organisms in this lab that grow only anaerobically. It's too hard to keep them happy, right? There's oxygen in this room, right? We had to set up those jars and it would be all kinds of fun that I don't have time for. Microaerophiles. What does micro mean? Small. So they need a small amount of oxygen. So we're going to find them where? Right? Near the oxygen zone, not necessarily in it. Right? In the pure oxygen zone. So which tube is that? That's E. You see how there's just a little bit of growth underneath the pink layer, but not anywhere else. Just underneath that pink layer. Facultative anaerobes. They can grow anaerobically or aerobically. So which one has growth throughout? Both A and B, right? See how it's cloudy all throughout? B is cloudy all throughout. But there's a distinctive difference between A and B, right? B has more growth up here at the top where there's oxygen and less in the anaerobic zone. We're no discernible difference for tube A, right? B is your facultative anaerobe, but facultative means the ability to change. These guys grow best in the presence of oxygen, hence more growth here at the top and less in the anaerobic zone. This guy grows strictly anaerobically. It's not utilizing oxygen. So its growth pattern is consistent through the oxygen and non-oxygen zones. And as its name implies, aerotolerant, it can tolerate oxygen. Unlike our strict anaerobes where oxygen would kill them, these guys can tolerate oxygen. Oxygen can be toxic for some organisms. Now, we have, what do you think in this lab? I already said we don't have the anaerobes because they're too hard to keep happy. Who else would be kind of difficult to keep happy? Do you think we have any aerotolerant? That would be pretty easy to keep happy, right? Facultative anaerobes? That would be easy to keep them happy. Anaerobes, not so easy. Obligate aerobes? No problem. Plenty of oxygen in here, right? What about those microaerophiles? They need just a little bit of oxygen. Is that easy to control? No. Not so easy. So anaerobes, strict anaerobes, and microaerophiles, we don't mess with. Too hard to keep them happy. So when we talk about organisms using and not using things, for energy, most of our organisms are catabolic, which means that they're going to break down molecules to release their energy. Right? I always think about my cat when I was a kid and how it broke down my dad's recliner very well with her nails. Right? So catabolic is tearing stuff apart, right? Cats are really good at that. So I guess it's just as well that cat, right, is in the word. So when we break down that energy, then it can be stored, and then later on it can be used for anabolism, adding stuff back together, right? Building molecules. So we're always constantly recycling energy from one place to another. Although proteins and lipids can be used as a source of energy, most commonly organisms use carbohydrates, sugars, and break those down for energy. Do you guys remember the processes from biology that we use to break down? One of them has to deal with one of the sugars that we commonly break down, which is glucose. And the breakdown of glucose is commonly referred to as glycolysis. Then remember the cycle? Krebs cycle or TCA cycle or the citric acid cycle has multiple names, right? And then what comes next? We got electrons we got to move, so we use the electron transport chain. Oh, it's all coming back, right? So the most common is 6-carbon sugar glucose that we break down, right? So the first step is glycolysis. That happens outside of our mitochondria. But even for bacteria, they use glycolysis too. Right? They just don't have mitochondria. They do it all inside their cytoplasm. The Krebs cycle or TCA or citric acid cycle, which is inside the mitochondria for us. And then the electron transport chain, which is also inside the mitochondria, more specifically embedded within the membranes in the mitochondria. So the organism traps some of the energy, right, that it releases in the form of GTP, or you've probably heard of ATP, right? When they add those phosphates, 3 of them, when they add that last phosphate, they store energy, right? And that bond between those molecules is storing energy. How do you make that bond? You need energy. When you break apart that bond, you release energy, right? So this is one of the currencies, one of the ways we store energy inside our cells. So do bacteria do this, right? They use ATP or GTP. And these are modified nucleotides. Same thing you find in DNA and RNA, except it's got extra phosphates. So a small amount of ATP is produced at what we call substrate level phosphorylation. So we're adding phosphates right at the enzymatic level, right? As we're breaking apart these molecules, we're using enzymes to transfer that energy. Most of the energy that we produce, though, is due to the electron transport chain. Electrons taken from glucose and glycolysis in the Krebs cycle are transferred to electron transport chain, and that process is referred to as oxidative because of electrons, right? We're gaining or losing, in this case, electrons. And this produces the largest amount of ATP. Now, those electrons have to go somewhere. So if you're an aerobic organism, where do those electrons finally go? What molecule grabs onto them? If you're an aerobe, like ourselves, what do we need to be able to do cellular respiration? Oxygen, right? Aerobe refers to oxygen. So some organisms can do that. If the organism doesn't have the ability to use the Krebs cycle or the electron transport chain, then it has to rely on just glycolysis, just the first step. And a lot of times to regenerate some of the electron carriers from glycolysis, it's going to add electrons back to its products from glycolysis. And that produces acids, right? So what happens in our body under anaerobic fermentation of glucose? What do we produce? What acid? Come on, you guys know this one. What builds up in your muscles? Lactic acid. See, I knew you knew it, right? And that can be very damaging to our cells. Other products that microorganisms will produce under fermentation are alcohols, right? Ethanol probably being one of our favorite, right? Fermentation gives rise to that. Gases are sometimes produced, carbon dioxide and hydrogen gas. Right? And lots of different acids can be produced. And again, it just depends on the organism, what food you give it, and under what conditions. It'll determine how much it produces of what substances. So those 3 pathways is what we commonly refer to as respiration. Now there's 2 different types of respiration. If oxygen is the final electron receptor, we said that's aerobic respiration, right? And we actually form metabolic water in the process. The electrons and hydrogen ions are picked up by the oxygen and you form water. Anaerobic respiration is something other than oxygen. So if you say something is an anaerobe, unless they specify what's the electron acceptor, you know it's not oxygen. It's got to be something else. So some common examples used by microbes are sulfate, and they'll reduce it into sulfite or hydrogen sulfide, nitrate into nitrite, and then into ammonia. Others can take nitrate into nitrite into nitric oxide to nitrous oxide, and then into nitrogen gas. And so that sulfur smell that sometimes you smell that with rotten eggs and in our swamps, in sewers, in some well water, is the sulfur compounds produced by sulfur oxidizing bacteria. Right? Sulfur is what kind of smells like rotten eggs. Right? So if you have a lot of sulfur in your water, that's why it smells like that. So when I lived in Ponchatoula for a while, our well water was kind of stinky. It's not going to hurt you. The ones that deal with nitrogen are denutrifying bacteria. Some of these guys will help the soil by putting nitrogen in. The ones that produce nitrogen gas are not good. They're taking the nitrogen out of the soil and putting it back in the atmosphere. And again, these are going to be found in soil and in stagnant water. What's the bottom line? If an organism can grow on media that contains sugar but lacks compounds necessary for anaerobic respiration, right? So if they don't have what they need, then they can grow by fermentation, right? And that's going to be mostly what we're looking at with these organisms is fermentation. Aerobic respiration using oxygen is going to amount the most amount of energy. Okay? So you get the most energy. And then fermentation, the cool thing with that is that it produces a lot of byproducts that are pretty easily tested, right? Acids we can detect very easily with pH changes. Gases we can detect by trapping that gas. Alcohols we can run special tests to test the presence of alcohols. And that's some of the stuff you're going to read about for homework and that we're going to do next time, is test for some of these byproducts. So not all organisms can metabolize in different ways. Some organisms have just a few pathways they can use. They haven't evolved to do everything. Okay. For our facultative anaerobes, for instance, they respire aerobically or they ferment anaerobically. Right? When they can't do aerobic respiration, most of them do fermentation. Right? The aerotolerant anaerobes, they're metabolizing anaerobically, right? They're using anaerobic respiration. They have some final electron acceptor other than oxygen. So on this planet, right, it started out, we believe, without any oxygen, right? And then we evolved to the presence of oxygen from photosynthesis. Most of the cyanobacteria and the algae in our oceans are actually what produce the excess oxygen on Earth that make it possible for aerobes. Not plants. Plants produce oxygen, but they also use it. So they don't produce excess. It's actually the microorganisms, right, and the vast oceans that produce that. So then, we have these ones that adapted over time, and it became more advantageous to be able to also utilize oxygen. So that's your facultative anaerobes, right? They can grow anaerobically, but they can also respire aerobically. And then you have ones like us, we've evolved completely to the other end of the spectrum, that we can't survive without oxygen, right? We have to have it for our metabolism. The problem with oxygen, too, though, is for some organisms it can be toxic, okay? So it can form toxic forms and actually kill them. So this is just a dot graph, right, showing the different ones that we talked about. So in FTM tubes, the dots represent the growth. So in tube 1, you're only seeing growth at the very top of the tube. So what would we classify them as? These would be our obligate aerobes, right? Where would our microaerophiles be in the drawing? 1, 2, 3, or 4? Everyone agree 4? Right, so it's in the oxygen zone, but not right at the top. Obligate anaerobes. 1, 2, 3, or 4? 5. 2. I started mostly 2s. Aerotolerant anaerobes. 5, because notice growth is uniform, where 3, do you see the difference here? More growth at the top, so that one is our facultative anaerobe. Because remember, it can respire aerobically. So why is it important the media be fresh when we do our FTM tubes? What type of organisms are we trying to grow, or what question are we trying to ask? Can it grow anaerobically? All right, we want to know, can they grow anaerobically? So we're mostly looking for our anaerobes and our facultative anaerobes. What happened if we used old media? It would have a lot of what? A lot of oxygen, So who's not going to grow? Our anaerobes, right? Who would grow and grow the whole length of the tube and really confuse us if oxygen had diffused all the way down? Those obligate aerobes, right? We get inaccurate results. So the older the media, the more likely that more oxygen's diffused. It would negatively affect our anaerobes, right? Too much oxygen. But it would positively affect our aerobes, which need oxygen. Maybe even our microaerophiles. Really wouldn't have much effect on our facultative anaerobes, right? Because they're going to grow no matter what. Not hard to keep them happy. So, for our obligate or strict aerobes, do they require oxygen for their metabolism? They do, don't they? They have to have it or they're going to die. Obligate strict anaerobes, do they require oxygen? No oxygen for those guys. Microaerophiles, do they require oxygen? Yeah, but a little oxygen. Facultative anaerobes, do they require oxygen? Do they have to have it? No, they do not have to have it, right? It's not required. Aerotolerant anaerobes, do they require oxygen? No, they do not, right? Can obligate strict aerobes grow in the presence of oxygen? Yes, in fact, only oxygen, right? What about obligate anaerobes? Can they grow in the presence of oxygen? No, they'd be killed. Microaerophiles. Can they grow in the presence of oxygen? Yes, but it has to be a small amount. Your facultative anaerobes, can they grow in the presence of oxygen? Yes, they can, right? They can respire aerobically. They'll grow quite abundantly, actually. What about aerotolerant anaerobes? Can they grow in the presence of oxygen? Yes, as their name implies, aerotolerant. Who can grow without oxygen? Can aerobes grow without oxygen? No. Can strict anaerobes? Absolutely, that's the only way they grow. Microaerophiles, without any oxygen, will they grow? No. Facultative anaerobes. Again, they have anaerobic in their name, so they can do it, but not as well. What about aerotolerant anaerobes? Yeah. Right, and that's, again, why we see these different patterns. What they use, what they don't use, right? What's harmful for them. So think about, again, why are we doing this? We want to know, is oxygen required? Can they grow anaerobically in addition to aerobically? Right? So we inoculated our tubes. Our tubes created those different environments. We can tell by the growth pattern what our organisms are doing. Right? Our really only true choices in this lab are strict aerobes, facultative anaerobes, and maybe an aerotolerant anaerobe. Oxygen utilization will indicate what they're using for energy. Or if they are fermenting anaerobically. So our anaerobic jars, again, we didn't do the experiment. We're not going to. We don't have enough jars. But again, we run into this problem. Can you tell if the environment has oxygen or not? By looking at the air right now. Other than the fact that we're still breathing and alive, right? It's the only way to indicate there's oxygen. So what if it was anaerobic? If the environment that you were looking in at was anaerobic? Again, you're not going to be able to tell that there's no oxygen. So what we do for those jars is we use the dye methylene blue. And under anaerobic conditions, it becomes clear, colorless. Right? So you don't see anything. Under aerobic conditions, what does methylene blue normally look like to us? Blue. Right? So you can see in my jar, See the blue tablet? Is there oxygen in there? Oh yeah, right? It's not sealed up. Oxygen has gotten into that environment. Here, notice the tablet in the picture. It's clear. You see that? It looks white. It's colorless. It's not blue. So what type of environment is in that jar right now? Under those conditions the picture was taken. It's anaerobic. Right? Tablet's colorless. So therefore, it's anaerobic. Note this packet was left out of the anaerobic chamber on the bench top. The tablet turned blue, right? Because it was exposed to oxygen. Right? In this room, there's plenty of oxygen. So that's aerobic environment. That packet that you see is a gas packet. It comes wrapped in foil. We open it up. We stick it in the chamber. That starts a reaction. The reaction releases hydrogen gas molecules and carbon dioxide. The hydrogen gas binds with the oxygen gas and it forms what molecule? Hydrogen and oxygen. H2O, water. That's not a gas form, right? That's liquid. So that's how we remove the oxygen, we turn it into water. So notice on this jar, what do you see? You see condensation, right? That's the formation of the water, right? So you know that there's a reaction happening here in that particular jar. And again, because it's sitting in the incubator too. So if the environment is anaerobic inside the jar, how will this affect the growth of these different organisms? Well, anaerobes. That would be the whole purpose we're setting up this jar, right? Are they going to grow in an anaerobic environment? Absolutely. They're going to be happy. What happened if we stick an aerobe in that jar and set it up anaerobically? What do we just do to them? We killed it. They'll die. You'll get no growth. Your facultative anaerobes. They'll still grow, right? Just not as well. Not going to be so happy. They're going to be fermenting. Not going to produce as much energy, but they'll still grow. What if the environment inside the jar sprung a leak, right, and became aerobic? If we didn't have our indicator, if we knew what the organisms were ahead of time, right? So if you had anaerobes you stuck in this jar, and then you took them out, and it had sprung a leak, you would suspect this because what would happen to your poor anaerobes? They'd die, right? You'd have no growth. Your aerobes would, on the other hand, grow, right? And they're not supposed to grow in an anaerobic jar, right? Oxygen must have got in. What about your facultative anaerobes? They'll still grow, and actually they should have better growth than we expected, right? Because oxygen got in, so they should be able to grow abundantly. So for the experiment, we usually take a set of plates and put them outside the jar and a set inside the jar. So what's the environment inside the jar if we set it up properly? Anaerobic. Is there oxygen present? No. What's the environment outside? Aerobic. Is there oxygen present? Yes, otherwise we'd all be dead. Okay? So Pseudomonas will grow under anaerobic, under anaerobic conditions, and it will grow under aerobic conditions. So what will we classify this one as? Hey, a strict aerobe. What's up, Melissa? They're in 108, I think, if you're looking for your sister. Is she here today?
Yeah.
Yeah.
108?
108 by the bathrooms in the middle.
Okay, alright.
You're welcome. So it would be aerobe, right? Because it only grew anaerobically. Clostridium sporogenes Grows anaerobically, not aerobically. This would be an anaerobe. Staphylococcus aureus grows under anaerobic conditions and grows more outside under aerobic conditions. This is your facultative anaerobe. Okay, so I'm going to speed through this one. Pseudomonas, does this require oxygen for growth? Yeah, it's an aerobe. It only grew aerobically, right? So it would grow really well with oxygen. Now, Clostridium sporogenes, does this organism require oxygen for growth? No, we just classified it as an anaerobe, a strict anaerobe, right? So will oxygen affect the growth of this organism? Yeah, it would kill it. Your Staphylococcus aureus, does it require oxygen? This is our facultative anaerobe. Does it require it? No. But it grows best in the presence of it, right? It would grow very abundantly. So, for number 1, it grew under both aerobic and anaerobic conditions. So this is our facultative, right? Number 2, no growth under aerobic, growth only under anaerobic. So this is our strict anaerobe. Number 3, grew under aerobic conditions, did not grow under anaerobic. Strict aerobe. Right, these are the pictures from your book. These are pictures from an experiment. So Pseudomonas, we notice, has that green discolorization you guys noticed last time, produces pigments. So you can see growth on this plate. You can see growth on the 2 E. coli and the 2 staph plates, right? The plates at the bottom here were under aerobic conditions. The plates at the top were under anaerobic conditions. We see as we expect for Pseudomonas, right? Under anaerobic conditions, what happened to it? It died. But staph grew under both conditions like we expected because it's a facultative anaerobe. E. coli grew under both conditions because it also is a facultative anaerobe. Right? I think that's our last slide. Oh, no. Oh, this is from today's quiz. So an alternative to the anaerobic jar is a candle jar. As the name suggests, a candle is placed in the jar, the lid, and it's lit, and then the lid is closed, and the flame will extinguish once most of the usable oxygen has been removed. What gas, though, builds up when you burn something? Carbon dioxide. So which classification that we did not talk about today, which was on your homework and on your quiz today, requires high levels of carbon dioxide? Capnophile. Other one that would probably be happy under this condition is your microaerophiles, because a small amount of oxygen does stay behind under that situation. So, scientific method again. That should have been our last slide. Yep. Let me make sure I do this